The overall goal of this procedure is to comparatively analyze the composition of multiprotein complexes in thylakoid membranes under different conditions. This is accomplished by first isolating thylakoid membranes from Chlamydomonas cells exposed to anaerobic conditions. Next, the thylakoid membranes are solubilized and fractionated on a sucrose density gradient. Then, equal volumes of the desired fractions are mixed and separated on SDS page. Finally, the Kumasi-stained bands are digested with trypsin. Ultimately, samples are analyzed by quantitative mass spectrometry to observe changes in protein abundance between the investigated fractions.
The main advantage of this technique over existing methods like quantitative proteomic studies comparing a defined amount of protein is that in our approach, equal volumes of fractionated samples are combined and analyzed. This allows studying the migration behavior of proteins within the gradient and moreover to analyze the composition of different multiprotein complexes in the thylakoid membrane with respect to the applied strains.
After culturing Chlamydomonas reinhardti strains according to the text protocol, use stock solutions of 2 molar sucrose, 10% beta-DM in water, and 0.5 molar tricine, pH 8.0 to prepare the following solutions for sucrose density gradients. To set up the gradients using 14 by 89 mm centrifuge tubes, begin by slowly pouring 1 milliliter of 1.3 molar solution, followed by 1 milliliter of 1.0 molar solution. Then pour 2 milliliters each of the 0.85, 0.7,